Welcome back to the deep dive. This time, we're traveling billions of years back in time
Oh, wow.
to explore the fascinating world of early microbial life. Imagine a world so ancient, it barely left a trace in the fossil record,
Right.
yet played a pivotal role in shaping the planet we know today. Intrigued?
I am.
We'll be using foundations for reconstructing early microbial life,
Okay.
a scientific review paper by Betul Kassar, to guide us through the very latest research in this field.
It's amazing how scientists are piecing together this incredible story using a combination of geology, bioinformatics,
Really is.
and even lab experiments that bring ancient molecules back to life.
It's like solving a mystery with only a handful of clues.
Hmm.
And the first challenge is the lack of traditional fossils.
Right.
I mean, we all love a good dinosaur skeleton,
Yeah.
but microbes are a whole different story.
It's not simply that they were tiny. Imagine trying to identify a microbe from a blurry billion-year-old photograph. Early microbes lack those distinct physical features that make other organisms easily identifiable through fossils.
Hmm.
So scientists turned to the field of molecular paleobiology,
Okay.
which analyzes ancient molecules like DNA and proteins preserved in rocks. This allows researchers to infer evolutionary relationships, metabolic pathways,
I see.
and even the environmental conditions of early Earth.
So like detectives, scientists are using these molecular clues to build a case for what life was like billions of years ago.
Yeah,
But how do we know the same rules applied back then?
that's a good question.
I mean, wouldn't early Earth have been dramatically different?
That's where the concept of uniformitarianism comes in. It's the assumption that natural processes we observe today, like the laws of physics and chemistry, have always operated in the same way. But can we apply this principle to the earliest life forms? To test this assumption, researchers use ancestral sequence reconstruction, which involves rebuilding ancient proteins in the lab and observing how they function under simulated early Earth conditions.
So it's like a time machine for molecules.
It is.
We're not just studying ancient genes, we're bringing them back to life.
Precisely. And what's fascinating is that while some ancient proteins behave just like their modern counterparts, others show intriguing differences, providing a more nuanced understanding of how life functioned billions of years ago.
This leads us to another big question.
Okay.
How did early microbes obtain and utilize carbon?
Hmm.
We know carbon is the backbone of all life,
It is.
but those early metabolic pathways are shrouded in mystery.
One approach is to analyze carbon isotopes. You see, carbon exists in two stable forms,
Okay.
carbon-12 and the slightly heavier carbon-13. Different metabolic processes prefer one isotope over the other, leaving behind a distinct isotopic signature in ancient rocks.
So by studying these carbon isotope ratios, scientists can determine what kinds of metabolic processes were active at the time.
Exactly. And one key player in this story is rubisco, the enzyme responsible for carbon fixation in photosynthesis.
Right.
It leaves a specific carbon isotope signature. By analyzing ancient rubisco alongside carbon isotope data from rocks, scientists are piecing together a fascinating picture of early carbon cycling.
That's remarkable. So we can trace the evolution of photosynthesis all the way back to these ancient microbes.
We can?
Were there other metabolic pathways important in early life?
Absolutely. Methanogenesis, the process where microbes produce methane, was likely prevalent on early Earth and contributed to the greenhouse effect that kept the planet warm enough for life to evolve.
Oh, I see.
Researchers are trying to pinpoint exactly when methanogenesis arose and its impact on the early atmosphere.
It's incredible how these ancient microbes influence the very air we breathe today.
Indeed. And it gets even more complex when we consider the possibility of protometabolic networks. These hypothetical networks predate modern metabolic pathways and might have been simpler and more robust, capable of operating under the harsh conditions of early Earth.
So not only do we try to understand the specific metabolic pathways of ancient microbes,
Yeah.
but we're also looking for clues about the origins of metabolism itself. Fascinating. But what about other essential elements?
Okay.
I know phosphorus is crucial for life as we know it.
Phosphorus is a key component of DNA, RNA, and ATP, the energy currency of cells.
Right.
But evidence suggests it might have been scarce on early Earth. This has led scientists to investigate the possibility that sulfur compounds played a more prominent role in early metabolism before phosphorus became dominant.
So early life may have relied on a different set of chemical building blocks.
It may have,
What are the implications of this?
well, for one, it could have dramatically impacted early Earth's atmosphere and oxygen levels. Understanding these early metabolic strategies is key to unraveling the interconnectedness of life and the planet's history.
Speaking of essential elements, how did early microbes access nitrogen, which is critical for building proteins?
Breaking the strong triple bond in nitrogen gas is no easy feat.
Right.
Yet the evolution of nitrogenase, the enzyme that does this, was a pivotal step in the history of life. There's an ongoing debate about when nitrogen fixation emerged.
Hmm.
Some geochemical evidence points to the existence of molybdenum-dependent nitrogenase over 3 billion years ago.
That's interesting, but didn't you say that molybdenum was likely scarce in the ancient oceans?
That's exactly the puzzle researchers are grappling with.
Okay.
How could molybdenum-dependent nitrogenase exist if the necessary element was scarce? And then there's the orphan G subunit in nitrogenase.
I see.
This subunit seems to have appeared after the Great Oxidation Event when oxygen levels rose dramatically.
Right.
Its function and relationship to the rise of oxygen remain a mystery.
Okay, so we've got carbon, phosphorus, nitrogen, all essential elements with fascinating histories. And then there's the incredible ability of some microbes to harness the power of sunlight.
Yes, the evolution of phototrophy, the ability to convert light energy into chemical energy, is a monumental chapter in the story of life.
It is.
It's believed that anoxygenic photosynthesis, which doesn't produce oxygen, predates the oxygenic photosynthesis we see in plants today.
So the first photosynthetic organisms weren't pumping out oxygen like modern plants.
Right.
How do they get their energy?
Recent research suggests the history of an oxygenic photosynthesis is more complex than previously thought.
Okay.
We're still discovering new lineages of microbes that carry out this ancient form of photosynthesis.
Uh-huh.
It seems anoxygenic photosynthesis didn't simply disappear with the rise of oxygenic photosynthesis. It continued to evolve and diversify alongside it.
Wow, so there were multiple paths to harnessing sunlight.
There were.
And then at some point, oxygenic photosynthesis evolved, leading to the rise of the cyanobacteria.
Precisely. Crown group cyanobacteria, the lineage we know for certain were performing oxygenic photosynthesis billions of years ago, ultimately paved the way for the Great Oxidation Event.
Wow.
This transformed Earth's atmosphere and set the stage for the evolution of more complex life.
It's a huge leap forward, but wouldn't oxygen have been toxic to many early life forms?
Absolutely. Oxygen is highly reactive and can be damaging to cells.
Right.
So alongside oxygenic photosynthesis, life had to evolve mechanisms to protect itself from oxygen's toxic effects.
Mm-hmm.
This is where superoxide dismutase, SOD, an enzyme that neutralizes harmful byproducts of oxygen metabolism, enters the picture.
Okay.
By studying ancient SOD enzymes, we gain a deeper understanding of how early life adapted to this dramatic shift in the environment.
So it wasn't just about producing oxygen. It was about learning to live with it. Fascinating. And speaking of fascinating, the paper mentions rhodopsin, a light-sensitive protein found in our eyes. What does this have to do with microbes billions of years ago?
Rhodopsin is actually found in all three domains of life: bacteria, archaea, and eukaryotes.
Really?
It's incredibly ancient, and while we associate it with vision in animals, it likely served other purposes in early life. The earliest forms of rhodopsin probably acted as ion pumps, moving charged particles across cell membranes.
So rhodopsin wasn't originally about seeing.
It wasn't.
It was about sensing and responding to the environment in a different way.
Exactly. And because rhodopsin is so widely distributed and its evolutionary history is encoded in the genomes of modern organisms, it's become a valuable tool for understanding microbial evolution and ecology, even when traditional fossil evidence is scarce.
I see.
We can glean insights into how early microbes sensed and responded to their environment, even in the absence of those detailed fossil records.
It's like using a molecular time machine to understand the behavior of these ancient organisms.
Yes, and it can reveal some surprising connections.
Okay.
For example, studying ancient rhodopsin variants can even provide clues about the behavior of ancient animals, including the ancestors of whales.
So from tiny microbes to the vastness of the ocean, from the origins of metabolism to the evolution of vision, it seems we're uncovering an incredible tapestry of life.
We are. And next, we'll delve into lipid synthesis and metabolism, which takes us into the realm of early eukaryotes, the lineage of life that includes animals, plants, and fungi.
Okay.
This is where things start to become a bit more familiar, but as you'll see, there are still many mysteries to unravel. Before we move on to early eukaryotes,
Okay.
I'd like to circle back to a point we briefly touched upon earlier, the protosteroid gap.
Right.
This refers to a period between 1.6 and 0.8 billion years ago, where we see a distinct lack of eukaryotic sterines, which are fossilized sterols. in the rock record.
That's right. A billion-year gap in the fossil record is pretty substantial.
Yeah.
What are the leading explanations for this?
One possibility is that early eukaryotes were simply rare during this time,
I see.
perhaps confined to specific environments that didn't favor fossil preservation.
Mm.
It's not that they disappeared entirely, but their traces are extremely difficult to find.
So like those early microbes we discussed.
Yeah.
The lack of fossils doesn't necessarily mean they weren't there.
Right.
It could just mean they were harder to detect.
Exactly. But there's another, more intriguing possibility.
Okay.
Remember those protosterols we mentioned earlier?
Yeah.
These ancient lipid molecules are not found in any living organism today. But their presence in ancient rocks hints at a very different lipid biochemistry in early eukaryotes.
So are we talking about a completely different type of cell membrane?
Potentially, yes.
Wow.
These protosterols suggest early eukaryotes might have built their cell membranes in a way that's completely foreign to modern eukaryotes.
I see.
This discovery has the potential to revolutionize our understanding of early eukaryotic evolution,
Okay.
forcing us to rethink what we thought we knew about the fundamental structure of their cells.
That's incredible. It's like finding a blueprint for a lost civilization, revealing a completely different way of building the foundations of life.
It really is. And this is a prime example of how studying ancient molecules, even those not directly involved in fossilization, can offer groundbreaking insights. By piecing together these molecular clues, we can begin to fill in those missing chapters in the history of life.
And speaking of uncovering hidden chapters, the paper also highlights how studying ancient proteins can offer solutions to modern problems.
It can.
It's a fascinating concept, but how can molecules that existed billions of years ago be relevant to the challenges we face today?
Think about it.
Yeah.
Life has been evolving on Earth for billions of years,
Right.
facing all sorts of challenges: extreme temperatures, changes in atmospheric composition, scarcity of resources. To survive, these ancient microbes had to be incredibly resilient and adaptable.
So their solutions to these ancient challenges are encoded in their genes and proteins, just waiting to be unlocked.
Exactly.
Yeah.
By studying these ancient proteins,
Yeah.
we can learn how life solved problems in the past and potentially apply those solutions to our own challenges.
What kind of applications are we talking about? Can you give us some concrete examples?
The possibilities are vast.
Okay.
For example, imagine engineering organisms with ancient metabolic pathways to create new biodegradable materials,
Right.
or to break down pollutants in the environment.
I see.
We might even find solutions for capturing and storing carbon dioxide from the atmosphere,
Okay.
drawing inspiration from the metabolic strategies of ancient microbes that thrived in a world with vastly different levels of atmospheric carbon dioxide.
So by understanding the past, we might find the key to unlocking a more sustainable future. It's a reminder that even the tiniest organisms can hold profound lessons for humanity.
I completely agree. And this leads us to the final chapter of our deep dive.
Okay.
By examining ancient life forms.
Yeah.
We're not just retracing evolutionary steps.
Right.
We're also grappling with some of the most fundamental questions about life itself.
What do you mean by that?
Well, as we've seen,
Yeah.
the early Earth was a drastically different place than our planet today. The atmosphere, the oceans, the very composition of the continents, all were in constant flux.
Makes you wonder how life as we know it even got started under such extreme conditions.
That's the core mystery, isn't it? Scientists are making progress in understanding how simple organic molecules might have assembled into the first protocells, but there are still so many gaps to fill. We're searching for those crucial transitions, those missing links that bridge the divide between non-living matter and the first self-replicating,
Right.
evolving systems.
It's a puzzle billions of years old, but it feels like we're getting closer to solving it all the time.
It does.
And it's not just about the origin of life, it's also about its incredible diversification.
It is.
Early Earth was a dynamic and challenging environment, yet life not only persisted,
Right.
it flourished and evolved into an astonishing array of forms.
We've only just scratched the surface of that diversity.
Yeah.
Different metabolic pathways, the evolution of photosynthesis, the emergence of eukaryotes. And beyond simply cataloging this diversity, we need to understand how it arose. How did life go from simple, single-celled organisms to the complex ecosystems we see today?
It's a question that goes beyond biology, doesn't it?
It does.
It speaks to the very nature of innovation and adaptability.
Absolutely. And then there's the intricate relationship between early life and the planet itself. We know microbes played a critical role in shaping Earth's atmosphere,
Let's think
oceans,
there.
and even its geology.
Hmm.
They were the ultimate ecosystem engineers.
Right.
But the details of that interplay, the feedback loops between life and the environment,
Yeah.
those are areas of active research.
So it's not just about understanding the evolution of life in isolation. It's about understanding the coevolution of life and the planet, a dance that's been going on for billions of years.
Yeah.
So to fully grasp the story of life,
Yeah.
we need to consider the entire stage on which it's unfolding.
We do.
It's a truly interdisciplinary endeavor, bringing together geologists, chemists, physicists, biologists, all working to reconstruct this ancient history. Yeah. What are some of the most exciting research avenues that are helping to answer these questions?
Ancient DNA analysis is one promising frontier. While recovering intact DNA from billions of years ago is incredibly challenging, new techniques are allowing scientists to extract and analyze fragments of genetic material from increasingly older rocks.
Wow.
This is giving us glimpses into the genomes of some of the earliest organisms, providing clues about their metabolic capabilities, their evolutionary relationships,
Right.
and their adaptations to the early Earth environment.
It's like eavesdropping on conversations from the dawn of life. What else is on the horizon?
Another powerful tool is the development of sophisticated computer models.
Okay.
These models allow researchers to simulate the conditions of early Earth, the atmosphere, the oceans, the temperature, the availability of various elements,
Uh-huh.
and then introduce virtual life forms to see how they might evolve under those conditions. It's like creating a virtual laboratory for studying the origin and evolution of life.
So by combining these virtual experiments with the insights gleaned from real-world data,
Yes.
we can start to piece together a more complete picture of how life arose and diversified on our planet.
It is.
It's a reminder that science is not just about collecting facts. It's about building a coherent narrative, a story that explains how we got from the simple to the complex, from the non-living to the living, from a barren planet to the teeming biosphere we see today.
That's a beautiful way to put it. And as we continue to explore the world of early microbial life,
Yeah.
I'm confident we'll uncover even more awe-inspiring discoveries.
Hmm?
Discoveries that will not only deepen our understanding of the past, but also inspire new technologies and solutions for a more sustainable future.
It's incredible to think that these microscopic organisms, so often overlooked in our everyday lives, hold such profound lessons for humanity.
They do.
It's a testament to the interconnectedness of all life, and a reminder that our own story, as a species, is intricately woven into the grand tapestry of life on Earth.
I couldn't agree more. If there's one thing I hope listeners take away from this deep dive, it's a renewed sense of wonder and curiosity about the natural world,
Right.
and an appreciation for the remarkable journey that life has taken on our planet.
Well said. And on that note, I want to thank you for joining us on this exploration of early microbial life. It's been a privilege to delve into this fascinating topic with you.
The pleasure was all mine.
And to our listeners, thank you for tuning in. We encourage you to keep exploring, keep questioning, and keep marveling at the wonders of science. Until next time, keep diving deep.